Welcome dear students to another very important metabolic pathway related to the glycogen metabolism, and that is the glycogenolysis. The word glycogenolysis means lysis of the glycogen, and lysis word refers to breakdown. So the literal meaning of the glycogenolysis is the breakdown of the glycogen. This can be defined as the breakdown of the stored glycogen to the glucose units. My dear students, glycogen, which is also called as the animal starch, it is the stored form of carbohydrates in most of the animals. And when the glucose is demanded in the body, at that time a process, a catabolic process takes place in the body that breaks down the glycogen to the glucose units. The glycogen is stored in liver cells, which are called as hepatocytes, and muscle cells that are called as the myocytes. So basically, glycogenolysis also takes place in the hepatocytes and the myocytes, and at subcellular level, the site for the glycogenolysis is the cytosol or lysosomes. It is a catabolic pathway in which there is no need of the energy in the form of ATP or UTP. So no ATP or UTP are utilized in the glycogenolysis. My dear students, this process is very important during emergency situations, or you can say fight or flight situations. Suppose a person is involved in a very intense muscular exercise, and for muscular contraction the ATPs are required. Then my dear students, the glycogen reserve of the muscles, they are broken down into the glucose, and that glucose through glycolysis produces the ATP required for the muscular contraction. And in case of the prolonged fasting, the fasting like for 12 or 18 hours, then the glucose level in the blood starts decreasing, and in order to maintain the healthy blood glucose level, then the glycogen reserves of the liver, they are broken down into the glucose, and that glucose is transported to the blood to maintain the healthy blood glucose level and perform the normal activities of the life. And this glucose is especially very important for those cells that only depend on glucose for their energy, like brain cells or the red blood cells. Now, my dear students, let us see how this metabolic pathway from the glycogen to glucose proceeds. This one is a glycogen molecule. You know, glycogen is a tree-like structure in which the glucose are attached both in the linear as well as in the branched fashion. When the glucose molecules are attached in a linear fashion, then there is alpha 1,4 glucosidic linkage between them. This linkage is present between the carbon number 1 and carbon number 4 of the two glucose units. But when the glucose units are attached in the form of a branch, then there is alpha 1,6 glucosidic linkage, means this glucosidic linkage will be present between carbon number 1 and carbon number 6 of the glucose molecules. So this one is the glycogen. First of all, there will be an enzyme, that is the glycogen phosphorylase. This enzyme is usually inactive, but once it is phosphorylated, then it becomes active. And when it becomes active, it starts the phosphorylated cleavage of alpha 1,4 glucosidic linkages. And when the alpha 1,4 glucosidic linkages are broken down, then my dear students, the catabolism starts, and there is subsequential removal or breakdown of the glucose units from the glycogen molecule. And my dear students, it is phosphorylated cleavage because this cleavage takes place when the inorganic phosphate attacks the molecule. And when this phosphorylated cleavage takes place and the glucose subunits, they get detached from this glycogen, then the free glucose is not produced, rather the glucose 1-phosphate molecules will be produced. And these glucose 1-phosphate molecules, then my dear students, there will be an enzyme phosphoglucomutase that will convert the glucose 1-phosphate into the glucose 6-phosphate, and then glucose 6-phosphatase will convert the glucose 6-phosphate into the glucose units. So in the first step, the catabolic process starts, and the glucose units, they start separating from the glycogen structure, but they are not released in the form of the free glucose, rather they are released in the form of the glucose 1-phosphate. And this process keeps on going until the glycogen phosphorylase is 4 glucose residue away from the either side of the chain. This is the alpha 1,6 glucosidic linkage, and there are 3 sides around this alpha 1,6 glucosidic linkage. This glycogen phosphorylase will keep on breaking the glucose units until there are 4 glucose residue left on either side of the chain. Once there are 4 glucose residue left on either side of the chain, just you can see that this is the side of the chain where there are only 4 glucose residues left behind. Then there will be a debranching enzyme that is specifically called as alpha 1,4 to alpha 1,4 glucan transferase. This enzyme will transfer a trisaccharide unit to either side of the chain in order to expose the alpha 1,6 linkage. So it will transfer this trisaccharide unit. This one is the trisaccharide unit consisting of 3 glucose, and that will be transferred to any side of the chain. Here I have transferred this trisaccharide unit to this side of the chain, and you can see 1, 2, 3, 4. There were 4 glucose units, and after the transfer of this trisaccharide unit to this side, now there are 1, 2, 3, 4, 5, 6, 7 units. So in this way, this trisaccharide unit that is transferred to either side of the chain, and this will expose this 1,6 glucosidic linkage that is responsible for the branching of the glucose units. After the transfer of the trisaccharide unit to any one side of the chain, then my dear students, the alpha 1,6 linkage is exposed, and there will be an enzyme amylo alpha 1 to 6 glycosidase. This enzyme will hydrolyze this alpha 1,6 linkage, and it will release this single glucose molecule in the form of the free glucose that will not be released in the form of the glucose 1 phosphate. Here the glucose single unit that will be released in form of the free glucose, and my dear students, now the branching diminishes and an unbranched alpha 1,4 polymer is obtained. So in this way, this linear or unbranched alpha 1,4 polymer is produced. It is a polymer because it consists of large number of glucose units. It is unbranched as there is no alpha 1,6 linkage now, and it will be having only alpha 1,4 glucosidic linkages. Then my dear students, again phosphorylase takes part in the reaction, and an inorganic phosphate attacks the molecule. Again the phosphorytic cleavage of the alpha 1,4 glucosidic linkages takes place, and these glucose units, they start detaching from each other. They are basically now detached from each other. And then my dear students, every glucose unit after the action of the phosphorylase and after phosphorytic cleavage of this bond, the glucose units, they are not released in the form of the free glucose, rather they are released in the form of glucose 1 phosphate. This glucose 1 phosphate is then, through the action of the phosphoglucomutase, it is converted to its position isomer, that is the glucose 6 phosphate, as the phosphate group is transferred from first position to the 6th carbon. And my dear students, this glucose 6 phosphate then in the liver cells only, listen very carefully, only in the liver cells, this glucose 6 phosphate that is changed to the glucose by the action of the glucose 6 phosphatase. This enzyme breaks down the phosphate group from the glucose 6 phosphate, and the glucose 6 phosphate will be converted to the glucose. My dear students, but the conversion of glucose 6 phosphate to glucose takes place only in the hepatocytes, and once the hepatocytes, they will produce the glucose, this glucose will move to the extracellular space, and then that will be transported to the blood in order to maintain the healthy blood glucose level. But my dear students, this glucose 6 phosphatase, this enzyme is absent in the myocytes. As this enzyme is absent in the myocytes, so that is why the glucose 6 phosphate is not converted to glucose in the myocytes. Rather, this glucose 6 phosphate will be directly involved in the glycolysis, and that will produce the ATPs for the muscular contraction. So basically, the glycogenolysis in the muscle cells is only to provide the ATPs for the muscular contraction by the process of the glycolysis. But the glycogenolysis in liver, that is to maintain the normal blood glucose level in the body for the normal homeostatic functions, because glucose is the main fuel that runs our body, and there are some cells that only depend on the glucose for their functioning, like the brain cells and red blood cells. So it is very, very, very important to maintain the normal or healthy blood glucose level in the body. And that is why the liver will just break down the glycogen to maintain the blood glucose level, and muscles will break down the glycogen in order to get the ATPs for the muscular contraction. So it was all about the glycogenolysis. I hope so that this lecture will be very understandable for you people. In case you find any problem, you can ask in the comment section. I will answer that. Till then, take care.